Started. Is it on now? Yeah. So we're going to get started with this first symposium, which will be focused on heart-brain interactions. We really have an absolutely excellent lineup of speakers to kick this thing off. And we're starting really, really strong with Veronica Egger. I will just ask you to keep your questions until the end of the talk, since we are trying to time keep these talks. And then we will have, after each speaker, 3 minutes of questions. And with that, I will let you kick it off.
Good morning, everyone. So I don't know if this is on. Thank you very much for inviting me, especially to the organizers of the conference, and most of all to Antonio. So he basically contacted me, I think, about 2 days after our paper on interoception came out. This was super impressive. And I really hope that you will enjoy what I'm going to tell you. So I'm coming from Regensburg University, and Regensburg is a small town a little bit north of Munich. And my background is in physics, but we have been doing cellular neurophysiology in the olfactory system since over 20 years. So this is now our model system, which is the rat. That is also different from what most of you are doing who are working with humans. So in olfaction, odorant molecules enter the nasal cavity when the animal or the human is inspiring, and the odor molecules bind to olfactory receptor neurons, which are located here at the olfactory epithelium. And in the olfactory epithelium, you have olfactory sensory neurons, which then project into the brain, into the first station, the olfactory bulb, which is located here. And in the olfactory sensory neurons, you have about 1,000 different types of olfactory receptors, and each neuron expresses just one subtype of receptor in rodents, and I think it's the same in humans. And these neurons are interspersed in the olfactory epithelium, but then in the next level, everything gets sorted out. So all the neurons with the same receptor will project to the same location in the olfactory bulb, a so-called glomerulus. And each odorant actually can activate several subtypes of receptors. So you have now a distributed representation of the stimulus on the level of the olfactory bulb, and my lab is interested in how this representation is actually moved forward in the brain to the next higher station, to the piriform cortex, how this actually works. And we are also interested in oscillatory activity in the olfactory bulb. So ever since the 1940s, when Lord Adrian recorded from the hedgehog olfactory bulb, it has been known that there are two very strong network oscillations. One is a slow theta oscillation that is coupled to respiration with a stimulus, and the other is a fast gamma oscillation on the order of 40 to 100 hertz. And this gamma oscillation is something that the spiking of the principal neurons of the bulb, the mitral cells, who then project onto the olfactory cortex, the spiking of these cells is spaced in this gamma rhythm. So it has been argued that the slow theta oscillations contribute to some sort of latency coding of olfactory stimuli, and the fast gamma oscillations have actually been implied in fine odor discrimination. And my lab actually has now come up with some hypotheses that the neurons that we are interested in, the granule cells and mitral cells, basically provide a means by which distant receptor channels can interconnect and talk to each other and become synchronized in the gamma range. So that's why I've become really interested in these oscillations. But we have been working, or we are working in acute brain slices, and acute brain slices are not a good system to investigate oscillations, because in slices, of course, we reduce a lot of the connectivity. And so therefore, we were interested to work with a more intact system, and we teamed up with these people here, with Matthias Dutschmann, and he is actually interested in the respiratory brainstem, and he helped us to develop a semi-intact preparation of the olfactory system. So what you do, you anesthetize the animal, you kill it, you transect it, you remove the heart, and then you replace the circulation with a peristaltic pump. So you cannulate the artery, the aorta, and by this you then perfuse the brain tissue. And we use as a readout actually the phrenic nerve, and you know that there's a respiratory pattern generator. So we increase in the beginning of the experiment the pump rate until this bursting pattern of the respiratory pattern generator appears. And this tells us then that the perfusion level is right, and that the tissue also of the olfactory bulb will receive enough supplies. And so this is how this basically works. So it's a very reductionist approach. there is no heart, no lung, no central top-down inputs to the olfactory bulb. And I just want you to keep this in mind. And so we have been able actually in this preparation to record responses to odorants, so you can also apply sensory stimuli. And we also have been able to record both slow and fast network oscillations in response to odorants. And there was one thing that struck us very early on, that even if we just recorded spontaneous activity without the presence of a sensory stimulus, there would be slow oscillations in the theta range. And this is really weird, because theta before has been related to the respiratory rhythm, but there is no respiration. So we thought that maybe this means that there is some kind of intrinsic bulbar network resonance, that then when the organism actually respires, becomes basically tapped into by this respiratory rhythm. And so we wanted to investigate this more, and this is now the work of my postdoc Luna Shamal, who did all the experiments. And basically she was very able to also record these slow oscillations. She found that the slow oscillations have harmonics, which is something that is also characteristic of neuronal network oscillations. And when you look at the distribution of the fundamental frequency of these oscillations, this is around 2.6 hertz, so in the theta range, at least in our system. But after a while, we actually noticed that there's something really weird, because if you zoom in on these peaks, you will see that these peaks in the Fourier transform are very narrow. And this narrowness cannot be explained for a biological system. So this is much too narrow for network oscillations. And so we grew suspicious, and indeed we had to recognize that these pulsations, these oscillations were actually tied to the pump rate. So when you increase the pump rotation rate, you get also an increase in the fundamental frequency. And the factor of the increase is actually tied to the fact that this pump has 8 rollers. So in fact, this is due to the pulsation that is imposed by this peristaltic pump, which is of course bad news. So we also confirmed this by including a so-called wind castle in the perfusion system, which dampens the pulsations, and indeed you get then rid of the slow signal. So at this point, the story could have ended, because pump artifacts are a very common nuisance in physiology research, as many of you may know. But at this time, we already had 2 indications that there might be also some biology related to this. So when you render your system hypoxic by removing the oxygen from the perfusion solution, the oscillations will go away. And also we noticed that the mitral cells, the principal neurons of the bulb, synchronize their spiking to the slow rhythm. So there must be some biology. And actually then I started to look into the literature, and we found that these perfusion pulsations and the pressure that we record in our preparation are, of course, not exactly the same, but somehow similar to the heartbeat-induced pulsations in living organisms. So here you have the aortic pulsation recorded in a mouse, and this is what you all know, the intracranial pressure pulsation in humans due to heartbeat. And so with this, I convinced my colleagues in Hamburg, who actually do in vivo recording from awake mice, to check whether there is an association between the heartbeat and neuronal activity. And so they recorded the ECG from awake mice. At the same time, they recorded for many neurons in the olfactory bulb using a multi-unit recording electrode. And in these recordings, they actually, first of all, found a very strong relation to inhalation. As all of you know, this activity in the bulb is strongly related to inhalation. But also, with a lot of averaging, they found that a subset, about 10% of the neuronal units, would actually be synchronized to the heartbeat. And so they also could confirm this in the cumulative data analysis. So about 10% of the neurons synchronized to the heartbeat, and they do this within a rather short latency of about 20 milliseconds. And so why has no one found this before? This is kind of weird, but the reason is that you really need to do a lot of averaging to find these effects. So they are rather weak. So Sebastian did averaging over tens of thousands of heartbeat, alignment of spikes, and then you will see this effect. So what could be the basis of this effect? So we were hypothesizing, because this is so fast, it has to be something that is actually located in the mitral cell membrane. And luckily, there was some literature showing that piezo2, a mechanosensitive ion channel, is expressed in a subset of mitral cells. So there were various papers coming out a bit just shortly before we got our results. And you know all that piezo1 and piezo2 are mechanosensitive ion channels that have been discovered only about 10 years ago. And for this, the Nobel Prize in 2021 was awarded to Adem Patapoutian. So there is, and we also found evidence from our recordings that the oscillations are actually originating from the mitral cilia, not from above and not from beyond. And so there is pharmacology for piezo2 channels. It's not perfect because this pharmacology also blocks TRP channels and piezo1 channels. It's a spider venom, it's from this tarantula, and it's called Grammostola mechanotoxin 4. And when you inject this toxin into the bulb, you will get a reduction of the oscillation. The control experiment is that if you inject lidocaine, which is a blocker of voltage-gated sodium channels, and which is commonly used to block neuronal network oscillations, you don't get an effect on the oscillation, but you get an effect on the overall activity in the local field potential, as you would expect for a sodium channel blocker. Oops, I'm sorry for that one. Okay. So now we have already additional evidence because we also silenced piezo2, though this is not in the paper. We silenced piezo2 with silencing RNA, and we get the same result, that we can get rid of the oscillations. So in summary, we have shown that there is some relation between heartbeat-induced pulsations and neuronal activity in the olfactory bulb. But what is probably more interesting for you is that in this paper in 21, they showed that the piezo2 channels are also expressed in principal neurons in other regions of the brain, for example, in cortex, in hippocampus, and also a lot in Purkinje cells in the cerebellum. So Sebastian also recorded from hippocampus and prefrontal cortex, and they also found entrainment of similar amounts of neurons to the heartbeat there, fast entrainment. And just to compare this to the classical ascending baroreceptive pathway that all of you know, we now have discovered a fast interoceptive mechanism that probably can exist in all brain areas with sufficient piezo2 expression and not just in the central autonomic network. It's very fast. As I showed you, it's about 20 milliseconds in red. So this pathway is multisynaptic, so it will probably take hundreds of milliseconds to actually reach the brain. But I've also shown you that our pathway is probably rather weak and modulatory compared to the synaptic pathway, which can really drive spiking of neurons. And what this all is about, the function, I have no clue. So I'm basically here at this conference to learn from you how this could actually contribute to cognitive processing. So this is just a couple of different ideas, and I guess you know these ideas much better than I do. And here you can just see one of these very fancy MRTs, which actually can now visualize heartbeat-induced oscillations of the tissue in the brain. Thank you very much.
Are there any questions in the audience?
Thank you for this very beautiful detective work, so very impressive. So you showed on your other slide that there is, of course, we have a baroreceptive activity, right? And then we have this new finding from your lab. So can you speculate or just talk a little bit about how these two systems can interact, whether they would complement each other, whether they would be in some way competitive, like, or whether they would just work together?
Yeah,
I know that you probably don't have a data, but can you just maybe think about this?
I want to go back, but it doesn't work. Yeah, not really. I mean, I have, because I also, for this one, would need to know, for example, about the piezo2 expression in these areas of the central autonomic network. And I know that several labs are working on this, but I don't know the data. So if piezo2 is actually downregulated there, or if it's even stronger, I have no clue. So therefore, I don't really want to speculate, because I don't even know if it's present there.
Okay. Thank you.
Are there any other questions?
If no one has, I have another.
Yeah, then go right ahead.
No?
No, I don't think. Okay, Diego has a question.
No.
Great. Thank you very much. It was amazing. So I would like to ask you about this second question you put here about this. How do you imagine, or how do you conceive the possibility that this habit is triggering the activation of distant brain areas that to bypass the communication if it's directly communicating these two? So how this will activate an actual network as is written there?
Yeah, for this, we are also lacking the anatomical knowledge. So right now, I would find it super intriguing to know where this subtype of mitral cells are actually projecting to. So we are now only beginning to reveal that different types of principal neurons in the bulb have different targets across the brain. So it would be super interesting. And of course, the same thing also holds for these principal neurons expressing piezo2 in the other brain areas, if they have a special projection area. So at this point, we even don't know if this network exists, and we would really need to figure out the anatomical basis for this. Mm-hmm.
Okay, so then once again, thank you, Veronica, for that fantastic talk.